thank you as always to Bill and the entire Park family for continuing to do this every year. It's very important to my research and our efforts in trying to find better treatments for what is a very challenging and difficult disease to face. And that disease is glioblastoma. And unfortunately, I think I've shown you this slide every year for the last 6 years, identifying that really we have not moved the needle very much with glioblastoma. And there are lots of challenges to this disease. And really, the median overall survival is only about 16 months. And there have been some molecular studies that are ongoing that have identified particular markers that can adjust those numbers. And Dr. Brastianos will talk a little bit about that and some of the challenges we face even in the context of identifying molecular markers that predict how individual tumors are going to behave. And really, we only have 2 drugs that have been approved for glioblastoma in the last 13 years. And so very little has changed, unfortunately, in this disease. So we're trying very hard to make an impact on this. So what I've been trying to do for the last few years is to share with you some of the research that I do and share what your efforts are doing to help support that research and talk about some of my research. And then I bring with me an invited speaker who also shares how the extended research in this field is also being done. So this is a clinical trial of bavdegalutamide, which I presented to all of you last year. And it's for patients with newly diagnosed glioblastoma. And bavdegalutamide is a very interesting novel agent. It's an immunotherapy agent, but it works in a very different way. It works both on the tumor cells itself, but it also works on the microenvironment of the tumor. And it tries to wake up the immune system so the immune system starts to recognize the tumor as foreign. And so this is a very interesting clinical trial that we're doing. And so far, we've now actually completed accrual. And we're waiting for the data to mature to see if we're really able to make an impact on the outcome of the disease. And so a lot of the efforts that you guys have been putting into has helped us to support this clinical trial. I'm very excited because this trial, the preliminary results were accepted for an oral presentation at one of our big national meetings end of this month at the Society for Neuro-Oncology meeting. So that's a recognition of the importance of this trial and some of the work that you all are supporting. So that's sort of what have we been doing so far? What's sort of the next steps that can happen to help improve the care of patients with glioblastoma? And this sort of moves into this very hot topic nowadays, which is sort of deep learning or machine learning. And my area of research is in looking at imaging. And when we look at MRI scans, we often just sort of see a picture of a tumor, sort of the structural anatomical picture of what a tumor looks like. But an MRI scan actually can provide us with a lot more information than just what that physical appearance of the tumor looks like. And this gets to the concept of big data. And images are really more than just pictures. And so there are 2 areas that are really emerging out of this concept of radiomics and then this concept of deep learning. So radiomics is looking at an MRI picture and looking at the different intensities of the picture, how bright are some areas, how dark are some areas, and that can vary across a tumor. You also can look at the shape of the tumor. Is it more elongated? Is it more round? And also in tumor texture, again, sort of how different are the different intensities and different colorations of the tumor across a different picture. And that's something the human eye can't pick up very well. We see that there are changes in the MRI scan, but quantitating that is very challenging. And so there's this concept of radiomics that can pick out those individual features on the MRI scan that may provide additional information about how a tumor behaves and how a tumor acts. And so we will be able to learn about tumor biology by looking in more detail at these individual MRI scans. And then the other area that's really very hot right now that my lab is starting to work on is in deep learning. So in deep learning, you take large data sets of information and you feed it into an algorithm. And the algorithm learns what is abnormal and what is normal and can give you information about what a new set of information that is fed into it it'll be able to predict how that information or how that tumor might act over time. And so these are very interesting areas of research to help humans understand all of this information that's packed into an MRI scan. So one of the things we've been able to do in my lab is to try to look at predicting IDH status. IDH has become a very important molecular marker in glioblastoma and other kinds of primary brain tumors. That's a very important prognostic feature. So if you have an IDH mutation, that means your tumor is going to be a better actor. And that would be very useful information to know before the surgeon goes in and plans their surgery. And so we've been using MRI scans and deep learning to predict whether or not patients have this IDH mutation status. And so we've been doing a pretty good job of being able to predict prior to someone going to surgery and knowing whether or not they have that IDH mutation status or not. And that's going to help the surgeon plan their surgery in a more accurate and better way. One of the other challenges that we face in brain tumors is that fundamentally we need to be able to measure tumor response. And the way we measure tumor response is by measuring the size of tumors on MRI scans. And that can be a very laborious, time-intensive process for a human to do. The other challenge is that humans will measure tumors in different ways. So the way I measure a glioblastoma on an MRI scan may be different from how Dr. Bastianos measures a glioblastoma on an MRI scan. That interjects a lot of human variability, a lot of bias in how we measure response to drugs over time. So it would be ideal if we could eliminate the human variability, the human element, the human interpretation of these scans, and have a more automatic process that's unbiased in how tumors are responding. So we've developed a program called Deep Neuro that will automatically segment brain tumors on MRI scans. And this is an open source program, so it's posted on the internet, and so anybody in any academic center can actually download this software program and be able to outline their own MRI scans and be able to use it for clinical trials, which would be a great advance because then we can, in real time, measure tumor response, which is a great advance. So to summarize a little bit what I talked about, machine learning, really the goal is to aid clinicians in more accurately measuring tumor size, which is sort of the cornerstone of how we determine if novel drugs are effective. We also can use MRI scans to provide more information than just tumor size, which is really what we've been using them for to date. And this gets to the concept of radiomics, which I talked about to predict tumor behavior, and then even radiogenomics, which is a lot of the area where Dr. Bastianos and I are collaborating with, looking at genetic changes in the tumor and seeing if we can identify those areas or how the MRI scan reflects genetic changes in the underlying tumor to provide prognostic information on individual patients. And these are really critical tools to incorporate into clinical practice and into clinical trials because it'll speed our assessment and determination as to whether or not novel drugs are working or not working, which is really what we need to do in this field, is to find and identify more quickly drugs that are more effective. So this is my lab, which is it's housed over in the Martino Center of Martino Center for Biomedical Imaging, which is a joint venture between MIT and MGH. And so these are all I feel like each year the number of pictures is growing, which I think is a good sign. But these are all the wonderful people who are working on this project. So I think it's very nice to see some of these are graduate students over here on the right. And so it just is a sign of the excitement and the fact that there are a lot of new blood and a lot of interest of people getting involved in this field. And it's been a great joy to encourage the next generation of investigators to become involved in managing this disease. So thank you for all your support. Maybe I'll let Dr. Bastianos talk now.
So what my lab does is we're trying to understand the genetic changes in cancer, and we focus on primary brain tumors such as glioblastomas. Oh, thank you. Yeah, and then, and as well as other types of primary brain tumors and metastatic brain tumors. So I'm not going to go into the introductory slide since Elizabeth already did that, but we know that glioblastomas are an unmet clinical need, and we really need to do better. And the important point I want to make is that targeted therapy has not been effective for glioblastomas. So targeted therapy, have people heard of targeted therapy here? Precision medicine, do we know what that is? Personalized medicine, so for those who haven't heard of it, it means we can identify which patients will actually benefit from targeted therapy by looking at those genetic changes in specific tumors and then targeting appropriately. So targeted therapy has worked in many types of cancers, and there are different targeted therapies. So these are just some of just long names, ignore the long names, but these are just some of the drugs that have worked in breast cancer, lung cancer, leukemia. So the question is, do they ever work in brain tumors, in primary brain tumors? So the question is, do brain tumors have targetable mutations? Meaning, so do people know what mutations are? So mutations are changes in DNA, and changes in DNA can drive cancer. They can drive primary brain tumors, they can drive metastatic brain tumors. So the question is, can we target any of those mutations? So what we've been doing in my lab is really genomically characterizing brain tumors to try to characterize those changes that are driving those tumors, to look for novel targeted therapies. So we've been focusing on other types of primary brain tumors, as well as we've done some work in glioblastoma in collaboration with Elizabeth. But let me just give you an example of precision medicine in action. So this is actually work that came out of our lab where we actually studied another type of brain tumor called a craniopharyngioma. Long word, hard to pronounce. They're rare tumors. They occur in only 300 cases per year in the US. So they're extremely rare, but they occur in children, they can occur in adults. And when patients have them, they have lifelong morbidities. They have lifelong difficulties, including they can have visual changes. Their pituitary glands cannot function properly. And they can be neurologically devastated, not only from the tumor itself, but also from the treatment of the tumor, from surgery and radiation. And no effective medical therapy has ever been, there's no effective medical therapy. People have looked at other therapies that have worked for other tumors, and they've tried them in craniopharyngiomas, and nothing has ever really worked. So this is just one example of a very large tumor here. And these tumors can compress the optic chiasm, compress the pituitary. So they're tough tumors. So work in my lab from a few years ago, we did pretty comprehensive genetic characterization to try to understand the mutations. And we actually found, so there are two subtypes of this tumor. So this is a mouthful, adenomatous and papillary. I'm going to quiz you on these after you've had a couple glasses of wine to see if you can actually pronounce this. But we found that 95% of the adenomatous craniopharyngiomas have this type of mutation, and 95% of this subtype has this particular mutation called BRAF. So for those of you who've never heard of BRAF, so BRAF is actually a target in other tumors. So melanoma patients, about 50% of melanoma patients have what are called BRAF mutations. And BRAF inhibitors have really revolutionized the management of melanoma. So BRAF inhibitors are targeted therapy. So we talked about precision medicine target therapy. BRAF inhibitors are target therapy. So this is actually a landmark paper showing this patient. So all these spots here are not normal. This is melanoma. Patient had BRAF. This is when BRAF was discovered. The BRAF inhibitors were discovered in melanoma. You can see that the tumors are melting away here. So we happen to find that 95% of this specific subtype of brain tumors have this mutation. So around the same time that we made that discovery, we had a patient who came into MGH requiring 5 surgeries in the course of 2 months because this tumor just kept coming back. And he kept coming back, and he was extremely sick every time he would come back. And the surgeons would go back in and remove the tumor, but it would keep coming back. And at that time, because we had just discovered BRAF in this tumor, in this craniopharyngioma, we tested him for BRAF. And so this is the patient here. This is his MRI. So this is if you're looking at a scan this way. And I'm sure Elizabeth has gone through MRIs with you already, but this is if you're looking at the brain this way. And this is actually a big tumor. So you can see this white here, not normal. That's not good. So this patient, this is the 5th time he came in with this very large recurrence here. So at that time, the neurosurgeons came into our office and said, we need to do something different for this patient. So we tested him for BRAF, and he had the BRAF mutation. So then we called the drug company, convinced them to give us the drug, because this is obviously off-label. It's not, it's not an indication because no one had ever tried it. And then by day 17 of therapy, his tumor was shrinking. And then by day 34 of therapy, you can see here, so lots of white badness here, and then the tumor is disappearing here. So by day 34 of therapy, his tumor was melting away. So based on this data, we've now started a national clinical trial that's open at multiple sites throughout the US to look at this inhibitor. And this is actually a trial. So Elizabeth is the imaging chair on this trial. So we're looking at the imaging characteristics. We're correlating with the genetics. So our lab is doing the genetics. Elizabeth is doing the imaging to actually follow these patients and see how they're going to do. But it's a really exciting trial that's a national trial right now where we're actively enrolling patients. So you can see we went from DNA to clinical trial to now bringing in the science, so bringing in more science and imaging and merging it all together. So that's an example of a primary brain tumor where we actually do see that precision medicine works. So now the question is glioblastoma. So have I confused you all yet? We're all following? Okay. Okay. All right. So now, so why haven't targeted therapies worked for glioblastomas, right? Why do we still have a lot of work to do for glioblastoma? So this is work, again, that we've done in collaboration to study glioblastoma. So using some of those same techniques that we've used for the craniopharyngiomas, and actually we've done some other work in meningiomas, but using the same techniques, we're now analyzing glioblastomas. So this is a paper that we published together where we actually looked at the genetics of glioblastomas to try to understand, do glioblastomas have targetable mutations? Do they have mutations that we can target with targeted therapies? So this was a study that we published last year, and we actually have a brain donation program at Mass General Hospital where patients towards the end of their lives are actually gracious enough to say that once they pass away, they want to donate their brain for research. And actually it's been incredibly rewarding for the patients and their families because they feel like they're contributing. And also, we're extremely grateful for patients who can do this towards, who can say that they want to do this towards the end of their life. So, so we use these tumor samples to analyze to see what are the genetic changes in glioblastomas. All right, so I'm about to confuse you all, but I'm going to walk you through this. So what we do is we take the original glioblastoma sample that was taken out from surgery at the initial diagnosis, and then we look at the glioblastoma once after the patient has passed away. And we try to say what's different between the before treatment and after treatment, what changes, and do we see targetable mutations in the glioblastoma? So here's an example. So what we do is we map out the evolutionary relationship, meaning we try to understand, number one, what are the mutations in glioblastoma, and number two, how do they change over time? So here's one example of a patient who had a targetable mutation. So this is a very complicated figure here, but here we looked at the sample before the patient, when the patient got surgery, and then after treatment. And the important point is that we found a targetable mutation in this patient, but then after treatment, the patient lost that targetable mutation. It was no longer there after treatment. So the glioblastoma is evolving, and it evolves quite quickly. So an analogy is, so Charles Darwin in 1837 mapped out branched evolution in his notebook back, 200 years ago, or in 1837, and showed, in the theory of evolution, showing the evolution, this phylogenetic relationship. So lots of branches, lots of evolution. In glioblastoma, we see this kind of evolution where mutations are happening, new mutations are constantly happening. So we see lots of branches, and some of these targetable mutations that we see in glioblastomas are present later, or they're lost. So there's still a lot of work to do in glioblastoma. And actually, similarly, we saw, and I'm not going to walk you through this, but we saw similar changes. So the important point, I'm going to try not to confuse you here, is we actually then looked at all the different mutations in these samples to say, all right, across this cohort of samples, which ones have targetable mutations, when do the targetable mutations happen. And unfortunately, what we found was that many of the mutations that happen in the glioblastoma, right now there aren't good targeted therapies for those mutations. So we still have a long way to go to actually target those mutations. And those for which we do have drugs that target those mutations, they're only present in a small number of the glioblastomas, or they occur later, so it's often too late to target those. So that's the issue with finding better treatments for glioblastoma. So obviously there's a lot of work that still needs to be done, and this is the same theme, so I won't go through it. But I do like to use the analogy, so I'm Greek, so I always go back to my roots. So the tail of the serpentine monster Hydra, so for those of you who don't remember, so Hydra was this monster with multiple snake heads. And every time a snake head would be cut off, a new one would grow back. Eventually, Hercules killed the monster by attacking the neck here, by burning the neck of the monster. If we think of glioblastoma, it has multiple branches, multiple mutations. It's this mixture of these subclones. And unfortunately, if we start just cutting off one of these subclones, one of these branches, it won't get to the whole tumor. So right now, we know what these mutations are here, but we still have a long way to go to actually target those mutations. And that's an area, an active area of research at MGH, but also, the companies, we're hoping that drug companies are going to do more research in this area. So precision medicine fails in glioblastoma. So far, what we see is that every patient did have genetic changes, but many of these were not targetable with current therapies. Genetic changes that were targetable were only present in some cases or changed over time. So we need to identify therapies that target those genetic changes in glioblastoma. So that's an area, a very active area of research right now. So, so I started with showing you an example of target therapy working in one primary brain tumor. So there's hope for, many types of brain tumors, but we still have a long way to go for glioblastoma. And so, the support that you all provide, will definitely, hopefully, will be able to make some strides in this area. So how to find clinical trials for those who are interested, clinicaltrials.gov. If you want to find out more information about the research we do, here's my lab website. Elizabeth and I work really closely together, so it's been actually a very fun collaboration. So did I confuse everyone? Okay, good.
A new drug comes out. Can you maintain cultures of these mutations so before we have to stick it in a human, you can at least get a first reaction?
Yeah, it's a great thought. So actually one of the things that my lab does is we're actually culturing cells, but also developing mouse models too. So trying to see if we can, some of these agents, and actually we've gotten some drugs from the companies to try these drugs out in the lab too. That's exactly right. So for something like the craniopharyngiomas, which is the other tumor I showed you, because there is already a target therapy that works for melanoma, and because that same mutation that's in the melanoma we found in the craniopharyngiomas, actually there was enough data that we could just go straight to human clinical trials. But for glioblastoma certainly, and for other tumor types, there is a lot of work trying to do exactly that, where you can culture it and then also look at there's also some nice mouse models too, and you can explore that. So yeah, that's a great question.
To what extent does Mass General collaborate with other hospitals in these efforts, and to the extent that they share it, how fast do they share it with one another? Or are they so focused on getting the answer from their lab versus others to withhold information?
For one of our clinical trials, our meningioma trial, we have 500 hospitals that are participating in that trial. So they send us the tissue samples, we share the data back to them immediately, and then they treat the patient with that inhibitor. So the samples are sent to MGH because we do the testing there, but then we provide the information right back to the hospital. Our same thing with the craniopharyngoma trial. We have, we have more than 50 hospitals that are participating in that trial. Same thing, so we're all collaborating. And then even in our lab efforts too, so some of the work that we're doing, I think we have 40 or 50 hospitals that we're collaborating with. And again, we share data back with them. So it's very much a national, international effort because you can't do good science in isolation. You need to collaborate. So we have a great network of collaborators, local collaborators. So our labs work closely together, and then we each have our network of collaborators. And then, we also collaborate closely with pharma too, because they're developing these drugs. So it's definitely very much a collaborative effort.
I think as we move more to this concept of big data and machine learning that I was talking about earlier, you really need big data sets to validate algorithms and make sure that you're getting the right answer, and you don't want it just in one data set. You want it confirmed in other data sets. So there's a big push to do more collaboration and sharing data sets across sites.
And even some of the genomic efforts or the genetic efforts, the NIH has a publicly available database. So you can deposit your genetic data into that database, or the investigators can, and then other institutions can access it. So we actually deposit all our data, and then others can reanalyze it. They can merge it with their own data set. So that's really important because, again, doing things in isolation doesn't, you can't do anything in isolation, or you can, but you just won't make big strides that way. So it's a very important question.
The NIH is making a push, so if you're NIH funded, you tend to have to deposit your data in one of these central repositories.
Which is the right thing.
I used to take care of oncology patients, and I know many patients willing to join the clinical trial. So it's glad to hear that you've already put it in nationwide. And I'm wondering, do they just visit the website and they can get the treatment?
It's a good question because if you go to clinicaltrials.gov and you put in glioblastoma clinical trial, there'll be 100 plus trials that are listed on that website. And so it's really hard as an individual patient to know if you're a match for that trial. So it really becomes a conversation with your treating physician as to what works for you and what makes sense for you. But as a patient, you can drive yourself crazy looking at all the available trials that are out there and trying to figure out, should I go to San Francisco for this trial? Should I go to Oklahoma for that trial? Because everybody is doing their own individual research. So it becomes a conversation with your treating physician to see what works best with you. And I think all of us have our patients' best interests in mind. And hopefully if there is a great treatment that's out there, we have it available at MGH, or we work very carefully to have it set up and working at MGH. But it's very overwhelming as a patient to try to sift through all of this information and figure out what works for you as an individual.
Yeah, and there's certainly it is a big issue, and there's certainly advocacy groups that are trying to help with providing more information. But it is an ongoing challenge. As if you're being treated not very close to an academic center, then even access to some of these clinical trials is still a challenge. So it's definitely still a big challenge, I think, with oncology in the U.S. But clinicaltrials.gov does have all those trials, but as Elizabeth mentioned, it's extremely overwhelming for a patient to look that up. So I think having close conversations with your oncologist, but not all oncologists may not necessarily know about every trial in the country too. So it is a challenge.
Is it within the reach of a layman to understand how a targeted treatment actually works? Are there drugs that can actually differentiate one DNA structure from another? Or what does a targeted treatment really do?
It's a great question. Do you want me to take that? So tumors get changes in DNA, and there are specific what are called hotspot mutations, meaning when that change in the DNA happens, it makes a change to the protein that activates the protein a certain way. So it has a very specific change that makes the protein behave in a very abnormal way, or it can be an activating mutation. So the way targeted therapies work is many targeted therapies will actually target those specific abnormalities, but they can be dirty, meaning most drugs also have other effects too, and that's actually why patients get side effects from these drugs too, is because they can have, they may not necessarily only be targeting that specific change. However, when they're designed, the goal is to try to have them as specific for that genetic change as possible. So, and there's different ways to design drugs, but they try to target that specific change in that DNA. But again, they're not perfect either, so they can target other things also.
So because of the genetic changes, the tumors express proteins. They have sort of, on the surface of the cancer cell, there's a funny looking protein. And so the targeted therapy identifies that as a funny looking protein and says, oh, I'm going to say that's abnormal, and it attacks it. And as a product of that, it ends up killing the tumor cell is the idea.
The idea is that genetic change, when it happens, it causes the cell to just start proliferating and not. So usually your own cells will actually have their own signals that they'll stop growing, but then the cancer cell will keep the cancer cells will keep growing, and they have these specific changes that you can target that can try to stop the growth of that cancer cell.
Is the relationship between big pharma and these drug companies and you as research docs, has that evolved over time, and where do you see that going? I mean, is that going in a good direction? Is it better than it used to be? What would you say about that?
We work closely with the pharma companies. They make the drugs. So we work closely with them. They give us the drugs for the lab to explore in the lab, and then we bring them into clinical trials. It is often an uphill battle because many companies haven't traditionally supported brain tumor trials and brain, so my lab also studies brain metastases, so metastatic disease. But in general, in the past, it's been hard to get the companies to support that. So it is often an uphill battle. But we do need them, and they need us because they need us for the patients. So it, it's.
In the landscape of cancer, I gave you that number of only 12,000 cases of GBM each year. For a big pharma, that's not a big, opportunity for them. Whereas for lung cancer or breast cancer, there's a lot more interest in them investing in those areas. So in brain cancer, we do face a little bit of an uphill battle trying to get their attention, but I think that's changing a little bit because they can sometimes get orphan disease status for their drugs because we are a smaller, cancer type, and so there are ways that we're trying to work with them to get their interest.
I was curious as to whether or not, in addition to pharma, using CRISPR in some way to try and create genes that could be implanted to counteract some of the bad ones. Is there work being done in that area?
There's a lot of research using CRISPR technologies right now. We're still, it's still a very active area of research. We're not quite ready for prime time for using CRISPR to treat brain tumors, but it's definitely a lot of work right now in that area. So it's an exciting area. Yeah.
Is it helpful being here in Boston with all the stuff happening in Cambridge on the commercial side? I mean,
Definitely.
I would think that would be an extraordinary advantage. And, how do you tap into that community and not only the medical, immuno-oncology stuff and all that, but also, MIT you mentioned, I think, in terms of, big data and bringing that into the medical community. Is that bridge really there?
I think it is. I mean, particularly in what I do with this, the Martinez Center for Biomedical Imaging is a joint venture between MIT and MGH, and so there's a lot of interest in tapping into the academic resources that we have here, and then also partnering with pharma. And oftentimes it's not big pharma, oftentimes it's sort of startups and small companies with sort of one product that they're trying to develop, and is there a new avenue in brain tumors? Maybe that's a chunk that they can bite off and tackle. So I think we're really well positioned here to sort of, partner with those people. And, MGH tries to do outreach efforts and work with companies and set up meetings with industry to try to say, okay, well, this company's coming in, this is their product, do you want to meet with them? So we're very open to doing that. And I think it's very important to build on the research that the companies are doing, because there are lots of things that they can do that academic labs can't do. So it's an important partnership.
Yeah, we work closely with MIT for some of the genomics we do too. So we use some of the technologies they use, and it's easy to walk across the street to go to Novartis and talk to them and convince them to give us the drug we need, and just keep knocking on the door. So yeah, it's a really good research intellectual environment too, to not just academically, but the biotech. It's a really exciting place to be. It would be a hard city to leave. So yeah.
Wonderful.
Thank you, everyone.